Thank you, Mr. Chairman. First of all, I would like to say that I am not Linda Sahrawi. I am Professor Karim Adjou from the Veterinary School of Alfort, but the first author of this work is Linda Sahrawi. She is my PhD student. She is working in my lab, and she is coming from the National Veterinary School of Algiers. So my presentation is entitled Diagnosis and Molecular Characterization of Cryptosporidium and Giardia in Sheep in Algeria. As you know, Cryptosporidium and Giardia are a very important zoonotic parasites of worldwide distribution and of medical and veterinary importance. Cryptosporidium parvum is one of the major agents of neonatal diarrhea in young ruminants such as calves, lambs, and goats, kids. Giardia duodenalis is the most frequent intestinal parasitosis in the world in man. So let me give you some data about ruminant breeding in Algeria. Algeria is a country located in North Africa between Tunisia and Morocco. The most of the animals are in the north because the big part of the country is the Sahara. In this country, they had 2 million cattle, 5 million goats, and 27 million sheep. And this data come from the Ministry of Agriculture. So only 3 recent publications were reported on cryptosporidiosis in dairy farms, and no data are available on cryptosporidiosis and Giardia in small ruminants. That's why the purpose of this PhD study is to investigate the occurrence and genotype distribution of Cryptosporidium and Giardia in sheep in different regions of Algeria, but especially in the north of the country. The specific objectives are first to identify the presence of Cryptosporidium genus and Giardia duodenalis in small ruminants farms. Second, to identify the Cryptosporidium species and to identify the C. parvum subtypes because they are very important, especially the zoonotic ones. And we do that in order to estimate the zoonotic potential of C. parvum and Giardia duodenalis in lambs in Algeria. So 84 fecal samples were collected from different farms in the north of the country. Other samples were collected from calves, but the results of this study will be presented in the afternoon in the session of parasitology. And I will focus my presentation now only on lambs. All the animals have less than 3 months of age. Most of them were with diarrhea, and some of them were without diarrhea. The fecal samples were screened first by immunofluorescence staining using Merifluor Cryptosporidium Giardia kits. We can find that in Meridian Diagnostics laboratories. And after that, the samples were screened by PCR, nested PCR, using in order to identify the genus Cryptosporidium, the nested PCR used to analyze the small ribosomal subunit 18S And this was followed by the restriction fragment length polymorphism in order to identify the species. And when the species are identified, when it is a C parvum, we do the subtyping with PCR products and sequence analysis of the surface glycoprotein 60 kilodalton gene. We do the same protocol for Giardia, and Giardia cysts were identified by immunofluorescence. DNA extraction was done like as I mentioned before. And after that, fecal samples were collected, and the DNA was extracted and amplified by PCR using specific primers. The sequencing analysis was carried out to determine assemblage group, and especially zoonotic ones. So let me show you the results. This is our results on Cryptosporidium species. From 84 fecal samples, we had only 36 positive by immunofluorescence technique. It represents 58%. And after that, the positive PCR ribosomal RNA were, we had 21 positive using this PCR on 36 samples. And if you look to the results, on the 21 samples, we identified 16 C. parvum and 1 C. ubiquitum. Animals were infected by a single parasite species, and in this case, we have predominance of C. parvum. Let me show you now the results concerning the age and the diarrheic status of the animals. As you can see here, all the animals were diarrheic, and they have less than 4 weeks. Only 2 animals were no diarrheic, and they were the older one, 27 days and 37 days. These results are related, of course, with the Cryptosporidium parvum. If we look to the C. ubiquitum, we have here 5 animals. Their age is under 4 weeks. 4 of them were diarrheic, only 1 was non-diarrheic. Let me show you now the results of Cryptosporidium parvum subtypes. This table shows you the location of the farms, the numbers of the farms, the host animal, the number of the samples, the positive samples for Cryptosporidium using immunofluorescence, the identification of Cryptosporidium species. As I mentioned before, there is 2 species, C. parvum and C. ubiquitum. And now let me show you the subtypes found in this study. If you look to this table, we have identified 2 family subtypes. The first one is 2A, here in red, and the second one is 2D. And in the 2A, we have, in the 2A, we have 2 different subtypes. I want to stress that these results are very important because all these 2A and 2D subtype families are zoonotic. Let me show you now the Giardia duodenalis genotypes in lambs in Algeria. Remember, this is the table where there is the normal assemblage known in the world for Giardia duodenalis. Assemblage A is specific to humans and other mammals. B to humans and primates. C and D is specific to dogs. Assemblage E is found in hoofed livestock. Assemblage F is specific to cats. G in rodents. And H come from seal and ghoul. And sometimes we have a mixed assemblage such as A plus E. If you look to our results, we have the most important assemblage we found is assemblage E, which is specific to hoofed livestock. We have 6 here in the northwest of Algeria, and we have 4 here in the north center of the country. It is important to stress that 2 mixed assemblage A plus E were found, one in the northwest of the country and another one in the north center of the country. And assemblage A is zoonotic. So, in conclusion, I can say that our findings are very important because they show that Cryptosporidium and Giardia are infections are common in sheep in Algeria. This study confirmed the occurrence of Cryptosporidium in sheep and reported for the first time the first molecular description of Cryptosporidium in lambs. C. parvum is the dominant species in neonatal Cryptosporidiosis in lambs in Algeria. This result is in concordance with some results obtained in Europe. For example, in Spain or in Greece, they have C. parvum as a dominant species. But there is some differences in Europe. For example, if you look to Belgium or to Norway, C. ubiquitum is dominant. And if we look to other countries like Australia, China, ubiquitum is very dominant too. The most important finding here in our study is the circulating of C. parvum zoonotic subtypes families 2A and 2D in small ruminants farms in Algeria. Our findings suggest too that sheep isolates contained mixed Giardia infections A plus E. So sheep could be a potential reservoir for zoonotic Giardia genotypes A. And the next step of this work is to do the same design of work in goat kids. And as I mentioned before, some preliminary data were obtained in calves, and they will be presented this afternoon in a parasitology session. I would like to thank all our collaborators in France and other teams in France and our collaborators in veterinary school of Algiers in Algeria. Thank you very much for your attention.